Welcome back to another explainer. I am absolutely thrilled to jump into today's topic with you. Now, we usually think of DNA as the master script of life, right? Like this fixed, unchanging blueprint. But what if I told you that life doesn't just read the script exactly as it's written? Instead, it actually relies on this brilliant system of molecular margin notes that dictate exactly how and when cells perform their functions. Today, we are unlocking the epigenetic code and exploring how a really incredible technology called Whole Genome Bisulfite Sequencing, or WGBS for short, lets us read those margin notes in perfect detail.
So, you gotta wonder, how exactly does the environment change gene behavior without actually altering the DNA itself? I mean, this is the core biological mystery of epigenetics. It's the entire reason why a plant can suddenly adapt to a severe drought, or why organisms can evolve to survive in harsh new climates, all while their fundamental genetic sequence remains exactly the same. The secret? It all comes down to chemical modifications to the DNA.
Okay, let's dive into our roadmap for today. We're going to cover the epigenetic code, the gold standard of WGBS, compare a few methylation techniques, look at a really cool resilient rice case study, and finally, wrap up with the cDgenomics solution.
First up, Section 1. The epigenetic code, reading the margin notes of DNA.
To really grasp epigenetics, we have to start with a molecule called 5-methylcytosine. Now, you know DNA is made up of 4 standard bases: adenine, thymine, cytosine, and guanine. But sometimes, cytosine gets this tiny chemical tag. 5-methylcytosine is essentially a covalent linkage between a methyl group and the 5' carbon position of cytosine. Think of it like a molecular sticky note placed directly onto the DNA strand. It doesn't change the underlying sequence of letters at all, but it dramatically changes how the cell's machinery actually reads and interacts with that sequence, effectively regulating the gene's functionality.
And let me tell you, these tiny modifications do some serious heavy lifting. 5-methylcytosine is responsible for gene silencing, meaning it can literally turn genes off. It plays a pivotal role in transposon repression, basically keeping jumping genes from wreaking havoc, as well as the inhibition of repetitive sequences. Plus, it's absolutely crucial for embryonic tissue development. And what's especially fascinating for those of us in agricultural and evolutionary biology, it is a primary driver of environmental adaptation. It literally allows populations to dynamically respond to their environments over generations.
Moving on to Section 2, the gold standard, WGBS, or Whole Genome Bisulfite Sequencing.
So if these epigenetic margin notes are so important, how do we actually read them? Standard sequencing machines can't tell the difference between a normal cytosine and a methylated one. They're blind to it. We need the ultimate magnifying glass. The WGBS mechanism relies on this incredibly elegant chemical workaround. Step 1, researchers treat the genomic DNA sample with heavy bisulfite. Step 2 is where the magic happens. This treatment forces all the unmethylated, normal cytosines to convert into uracils. But the methylated cytosines, the bisulfite doesn't even touch them. They stay exactly as they are. Then, in Step 3, PCR amplification turns those new uracils into thymidines. Finally, Step 4 uses high-throughput sequencing. When we compare this completely new sequence to a reference genome, any cytosine we still see must have been protected by a methyl group. It's just a brilliant molecular transformation.
And the result of that process? You get single-base resolution mapping across the entire genome. We are not just getting a vague, zoomed-out idea of where methylation is happening. We are mapping it down to the absolute individual letter of DNA, literally everywhere in the genome. This extreme precision is exactly why WGBS is universally recognized as the unparalleled gold standard in the field of epigenomics.
Let's see how this stacks up in Section 3. Comparing methylation techniques. Why WGBS stands out.
When we contrast the most common laboratory techniques, the limitations of the others quickly become pretty obvious. Let's look at MeDIP-seq first. It uses antibodies to enrich methylated regions. It's good, but it only gives you regional resolution, not single base, and it only provides relative quantification, not absolute. Then there's RRBS, or Reduced Representation Bisulfite Sequencing. That one gives you great single-base resolution, but its scope is limited strictly to CG-rich regions. It completely misses the whole genome status, meaning you lose out on distant regulatory elements and gene deserts. But look back up at WGBS, single-base resolution, whole genome scope. It captures the absolute complete picture. Yes, the library preparation is highly complex, but the data you get out of it is just unmatched.
Let's move to Section 4. Case Study. Resilient Rice. Environmental Stress Response in Action.
Okay, let's take this complex technology out of the abstract and get it into the dirt. Researchers published a really fascinating study in Scientific Reports looking at 3 very different cultivars of rice. First, we have IR64, which is unfortunately highly sensitive to both drought and salinity. Then we have Pokkali, which is an absolute champion, highly tolerant to salinity. And finally, Nagina 22, or N22, which is highly tolerant to drought. The researchers isolated the genomic DNA from these specific plants and used WGBS to figure out why some survive extreme environments while the others just die off.
What they found was a direct, undeniable correlation between methylation and survival. They looked closely at differentially methylated regions, or DMRs, between the strands. Genes proximal to highly methylated DMRs were associated with lower transcript abundance. Essentially, the heavy methylation was acting as a switch to turn those genes off, down-regulating them. Conversely, lowly methylated DMRs were associated with higher transcript abundance, meaning the genes were switched on, upregulated. The plants were dynamically using methylation to literally flip their stress response genes on and off.
The researchers concluded the study with this really powerful statement. They wrote, DNA methylation plays an important part in abiotic stress response by regulating expression of a suite of stress-responsive genes in rice mostly via methylation or demethylation of proximal TEs. TEs in this case are transposable elements. The study actually proved that by modifying these specific transposable elements, the plants could sense and successfully adapt to massive environmental changes. It is a stunning, real-world example of evolutionary adaptation captured right at the single base level, and it was all made visible by WGBS.
Which brings us perfectly to Section 5, the CD Genomics Solution, expert partnership for your research.
Look, if you are planning to run an incredible study like that rice project, you probably already know that the sheer complexity of WGBS library prep and data analysis can be incredibly daunting. CD Genomics is a leading provider of these next-generation sequencing services, and they offer a totally seamless end-to-end workflow for WGBS. They handle the heavy lifting for you, starting right from step 1 with strict DNA sample preparation and quality control. They manage the highly delicate cytosine bisulfite conversion and tagging in step 2, they handle the PCR amplification and Illumina adapter introduction in step 3, and then they run the high-throughput, paired-end 150 base pair sequencing. But here's the best part, they don't just dump a massive hard drive of raw data on your desk. Step 5 is providing customized advanced bioinformatics analysis to actually help you make sense of the methylome.
To give researchers total peace of mind, CD Genomics actually provides guaranteed deliverables. They ensure that over 80% of bases will have a Q30 or higher quality score and that the sequencing depth will strictly exceed 20x. You receive the original raw sequencing data, comprehensive differentially methylated regions mapping, exactly like the rice study we just talked about used, along with detailed clustering analysis. And the real game changer is that they provide publication-ready data analysis reports, ensuring that this high-quality data immediately becomes a credible, comprehensive foundation for your own research publications.
So we've seen exactly how WGBS acts as the ultimate magnifying glass for the epigenetic code and how leaning on expert partnerships makes utilizing this amazing tech easier than ever. Which leaves me with one final question for you to chew on. What epigenetic mysteries could you unlock in your next evolutionary, agricultural, or biological research project by partnering with CD Genomics? The margin notes of life are out there, just waiting to be read. Thank you so much for joining me on this explainer. Stay focused, stay curious, and I'll catch you in the next one.